Okay, let's start. Hello, everyone. Thank you very much for joining us for today's molecular connectivity online series. This will be the last online series before the summer break, and we will have that. We will assume that in September. So my name is Cindy. I'm from New Jersey Institute of Technology. In case you are new here, so the molecular connectivity working group is a group of scientists aim to advance the molecular connectivity as a method to study brain connectivity. We organize this online series monthly to promote scientific research on molecular connectivity. For more information, you can visit our website and subscribe to our newsletters and follow us on Twitter or LinkedIn. Today, I'm pleased to introduce Dr. Tommaso Volpi. Dr. Volpi is currently a postdoctoral associate at the Department of Radiology at Yale University. Before that, he obtained his PhD in 2023 from the University of Padova, Italy, with the thesis on the complex coupling between FDG PET measures of brain glucose metabolism and fMRI proxies of spontaneous activity, including the comparison of metabolic and functional connectivity. His main current project concerns methods for PET kinetic modeling and non-invasive input function estimations to obtain physiologically information parameters and the integration of PET and fMRI-derived features to understand brain functions and connectivity. In today's talk, Dr. Volpi will cover molecular connectivity and dynamic PET, comparing time series and subject series approaches. And today, Ariana will moderate the discussion after the talk. So let's welcome Dr. Volpi. Thank you.
Thank you very much. Thank you very much for the introduction. Okay, so can you see my screen? I assume.
Yes.
Good.
Yes.
Okay, so thanks again, and good morning, everyone. Good morning, at least in the United States, on the East Coast. So I'm starting with this because the work that I'm going to present today is the work that I did during my PhD, that I started with my advisor, Professor Alessandra Bertoldo, and it's going to cover many of the discussions and of the work that we've tried to do to better understand this topic. And in many of these discussions, Dr. Erika Silvestri, who was a postdoc in Alessandra's lab at the time, was also involved. And now this work is being continued by Alessandra's students, and in particular by Giulia Vallini. And so I'm going to present some of her work at the end. And also I want to thank, of course, the Molecular Connectivity Working Group that I am a part of. And in particular, I'm a part of the Validation Council. And so this word validation will come back a few times during the talk. So what am I going to try to talk about today? So the main part of the talk is going to be our approach for trying to choose a way to derive a single subject PET connectivity estimates from dynamic PET data. And then I'm also going to talk about how we related these single subject connectivity estimates to across subject estimates, which are more frequently derived in the literature, and also to independent measures of brain connectivity, so structural and functional connectivity. And finally, I will conclude with how we try to apply this single subject PET connectivity approach to study brain tumors, in particular, to study gliomas. So let's start with the first point. And to start, I also need to do a brief introduction on PET quantification, what we mean by PET quantification. So in the clinical setting, typically what we do is we inject a PET tracer, for example, FDG, the tracer to track glucose metabolism, and then the patient waits a requisite amount of time, and the scan begins after the tracer has reached some sort of steady state. state, some sort of equilibrium. And one single image is reconstructed from the period of time that the subject is scanned, 10, 20 minutes, depending on the tracer. And then from that single image or static image, we get a semi-quantitative index of tracer uptake. Probably the most famous is SUV, the standardized uptake value. And then we have the SUVR, which is an SUV relative to a reference region. But what we like to do in research is actually to follow the tracer kinetics through the entire experiment. And so what happens is that images and PET counts are acquired from immediately after the tracer is injected, and for an extended period of time that can be 1 hour, 2 hours, depending on the tracer. And then multiple PET images are reconstructed. And so we're going to have a 4D dataset, a dynamic PET dataset. And then how can we analyze this dynamic PET dataset? Typically, the most accepted way to do it is to use compartmental modeling. Compartmental modeling is a mathematical framework to describe the tracer kinetics, which needs what? It needs the PET data, but it also needs an input function. So it needs that we need to know the tracer concentration in the arterial plasma, what you see here called the C with P. So armed with these two information, the arterial plasma tracer concentration and the dynamic data, we can estimate multiple kinetic parameters that describe the single steps of the tracer that the tracer undergoes in our tissue. So for example, here you can see images of K1 and K3, which are microparameters or single rate constants. Specifically, K1 is the delivery of the tracer into the tissue, and K3, in the case of FDG, is the phosphorylation of the tracer, the phosphorylation rate by the hexokinase enzyme. Or we can have parameters that collapse together, that lump together these single rate constants, like Ki, which is a combination of K1, K2, and K3. I also want to stress that this framework interprets the data, interprets the PET experiment as a linear time invariant system, which seems kind of complicated, but what I want to stress here is the fact that we get a single estimate of our K1 or our K3 or Ki for each brain region or for each brain voxel for the entire experiment. We don't get a time series of K1s or a time series of K3s and so on. Now, of course, this framework can be simplified with graphical methods or spectral analysis, so simplified analysis methods to obtain maybe a few of these parameters. Or potentially, we can also resort to equilibrium analysis if we change the way that we administer the tracer. In this case, I was describing the classical situation for a bolus injection, but we can also perform a continuous infusion. And so in that case, we can perform some sort of ratio between the tissue and the plasma, for example. And we're going to come back to this continuous infusion a couple of times. On the other hand, we have the field of brain connectivity. The field of brain connectivity has kind of revolutionized neuroscience because it interprets the brain as a network of nodes and links that connect those nodes. And in human experiments, in human brain imaging data, typically we have these types of brain connectivities. We can talk about structural connectivity, which is typically assessed using diffusion imaging, and which is a proxy for the anatomical connectivity between different brain areas. We instead have functional connectivity, which can be assessed with functional MRI, but also with electrophysiological measurements like EEG, which is typically a measure of the statistical dependencies between the time series of the signals of different brain areas. And we can also talk about effective connectivity when we try to add some directionality, some causality to the functional connectivity. And so we try to understand how neural populations interact with each other and influence each other causally. But what about PET? What can we do with PET in the field of brain connectivity? Now, typically, what happens, as we were mentioning before, is that for a given subject, we have multiple, we have for every region just a single value of a given parameter. It can be SUV, it can be Ki, it can be a binding potential, but we don't have a time series of these values, as we were saying. And so each subject in this matrix is a vector of region-wise values for that given parameter. And so to assess connectivity, we typically do it across individuals, across subjects. We can typically perform some sort of normalization of standardization to remove some of the inter-individual variability that we don't want. Of course, this can be more or less justified, but we can come back to that. And so then typically a correlation between these regional estimates is calculated across subjects. And so we have a final adjacency matrix, which is our group level across subject PEC connectivity matrix. We can then threshold it in some way, turn it into a graph, and analyze it using graph metrics, as is typically done in the functional MRI literature, for example. So this approach to across subject PEC connectivity is what many people call covariance, even though it's not covariance in statistical sense, because it's correlation, it's a normalized covariance. A slightly more complicated approach is to use sparse inverse covariance estimation, or SICE, which directly estimates a sparse graph. And we're going to come back to this. And these approaches are typically performed at the region, with the region level data. With voxel level data, people typically use the principal component analysis or independent component analysis. And you can see an example here of networks extracted with independent component analysis from VT images, so images of the total distribution volume of this tracer for synaptic density. But we come to the main question of this talk. So we get a group level estimate of molecular connectivity. How can we get an individualized estimate of molecular connectivity? And why do we care about that? We care about that because we want to understand how the brain physiology works, but also potentially how brain pathology alters these networks. And so potentially molecular connectivity could become a biomarker for some diseases. And of course, a biomarker requires an individual level estimate. Now, some authors have tried to derive an individualized estimate from subject series approaches, so from approaches obtained across subjects, and usually this entails evaluating how the single individual contributes to that group level network, and how removing that individual is going to perturb that network. Another possibility is, of course, to have multiple sessions for the same subject. And so to evaluate some sort of multi-session or longitudinal molecular connectivity network, but of course you have to have a given amount of these sessions to be able to make the network statistically meaningful. But the other natural option is actually to try to use the time series, so the dynamic PET data, kind of like what fMRI literature does with functional connectivity. So to use the time series of the bold signal to extract functional connectivity networks. However, this is potentially problematic for PET data, because here you can see the time activity curves from 4 different regions for an FDG data set. And if we just apply Pearson correlation, like we would do with functional connectivity with fMRI, what we get is a correlation matrix like this. So we get a correlation matrix which is full of very high positive correlations without a lot of without very much any network structure. And why is that? Because these time activity curves are very similar to each other. They all share is seeing the same shape, the same strong positive trend in this case, which in this case means what? It means that the FDG tracer is being accumulated in the brain tissue, and it happens for all the regions. And so is there a way that we can normalize this, that we can remove this trend, and should we even do it? So in the literature, a few studies had begun to tackle this question. So this was a paper by Tommaso et al. In 2017 that was working at the voxel level with ICA and seed-based correlation. And these were a few other papers that were working in the case of Amend 2019 on an animal model. And then we also have the paper by Sharna Yamadar in 2021 that was working on humans, using correlation approaches on region level data. And so in most of these works that were published before or at the time when we were studying this, to study this problem, some sort of standardization was applied. And in particular, the most popular was dividing the regional time activity curve by the whole brain average time activity curve. So to use that to detrend the PET signals. And so we started with a pilot study that was published for in a conference EMBC in 2021 with just 4 subjects from the Padova PET-MR, the Biograph MMR. FDG acquisitions, 60 minutes data. We used 2 different parcellations to assess the impact of an anatomical versus a functional parcellation on our results. And we were testing 3 different standardization approaches. In particular, the one that is most popular in the literature, and 2 others that combine demeaning and z-scoring to differentially emphasize the fluctuations of the signal. In particular, you can see that this second normalization is strongly emphasizing the fluctuations around the mean signal. And then we were doing kind of a very systematic approach. And so we tested multiple different methods that we classified as size and non-size. We were focusing on the size methods because, as we were mentioning, size induces sparsity. And so our idea was to try to tackle the strong similarity between time activity curves by trying to produce a sparse graph. The non-size methods instead were to have a more simple approach, less demanding from a mathematical standpoint. So very briefly, the size methods do what? They use a graphical lasso, so the lasso penalty, to introduce sparsity in the precision matrix, and then the precision matrix is converted into a partial correlation matrix, which is the final network that we use. Instead, the non-size methods that we were using were, of course, the Pearson correlation, cosine similarity, which is a slightly different flavor of similarity metric, but still both of these evaluate what? They evaluated the covariation in the signal fluctuations, in the relative patterns of the signals coming from 2 regions. And finally, Euclidean similarity, which is just a complement to 1 to Euclidean distance, which instead has a slightly different way to look at the signals, because it's assessing the similarity in amplitudes and in shapes of the 2 signals. And in particular, I want to focus on Euclidean distance because it's going to come back later. Euclidean distance is actually an approach that we typically use in PET at the voxel level when we do cluster analysis to identify regions that have some sort of biological meaning that we can then use for modeling. And as you can see here, the clusters that come out are not salt and pepper in pattern, but they identify regions that have some sort of biological relevance. And so, of course, we can estimate networks. But what's most important is, do they have any meaning? Do they have any biological meaning potentially? And so this slide on how to validate will come back a couple of times. So of course, the first thing that we can do, we can evaluate how reproducible these networks are across subjects, because we want some level of similarity across subjects. We can also compare them to independent measures of brain connectivity, like the ones that we can obtain from structural connectivity or functional connectivity. We can assess if these network measures correlate with cognitive tests or with neuropsychological variables that we can obtain for subjects, or we can potentially assess if they are altered by lesions in a biologically plausible way. And so here we started simple, of course, from the between subject reproducibility. And what I wanted to draw your attention to is the fact that the non-size methods actually have a much higher between subject reproducibility, irrespective of the normalization and irrespective of the brain parcellation that is used. And then we compared our matrices with a structural connectivity template obtained from Human Cognitive Project data. And again, we saw that the non-size methods had a higher similarity with the structural connectivity, which doesn't necessarily mean anything, because we don't want the functional network to be identical to the structural network, but still it can give us some hint that we're going towards a biologically informative network measure. And so this is the main study that we did that was published in Journal of Cerebral Blood Flow last year, with a much larger data set provided by Washington University in St. Louis, so 54 healthy subjects scanned with dynamic FDG. And between the approaches that we were mentioning before, we did not use size anymore. We focused on the non-size methods. And again, we asked, which is the metric that we think is the most appropriate? And do we need standardization of the signal? Do we need the trending? Yes or no? And so here you can see for Pearson correlation, in this case, this is the non-normalized matrix. Again, you can see that it's very high values and not a lot of network structure at all. And now we can see the single normalizations at the group average level. So here we have the time activity curves that were divided by the whole brain mean time activity curve. Here we have the second one with z-scoring across parses and the meaning across time points. This is the opposite operation. And you can see that for 1, 2, and 3, we have now some sort of network structure. We have some communities of regions. We also tried 2 other additional normalizations using the plasma input function, dividing by the plasma input function or by its integral, but they were not actually modifying the structure with respect to the non-normalized way that much. And here instead you can see the Euclidean similarity, the group average matrix, and I want to draw your attention to it because it's already showing some good network characteristics that we are interested in, because we have kind of a block diagonal structure along the main diagonal with communities of nodes that are more similar to each other with respect to the rest of the brain. And also, since we organized it with the left hemisphere before the right hemisphere, you can see these secondary diagonals that represent what in connectivity studies? They represent the connections, the similarity between regions that are homologous in the 2 hemispheres. So homotopic connections, which are typical of structural and functional connectivity studies. And this is the corresponding matrix for the Schaefer atlas. So you can see that there's consistency between the 2 atlases. Again, I want to stress that this is the non-normalized matrix. We are not applying any detrending to the signals. So then we did a slightly more quantitative assessment. So we calculated modularity on each of these average matrices for Pearson correlation and for Euclidean similarity. We did not do it for cosine similarity because the results were very, very similar to Pearson correlation. So I'm just showing it for these 2. And what we can, well, we want these networks to have a relatively high modularity, because we want to see that there are these communities of nodes that talk to each other more than they talk to the rest of the brain. And so you can see that normalization 1, 2, and 3 for Pearson have relatively high modularity, which is what we were already seeing qualitatively from the images. And for Euclidean similarity, we have that the non-normalized version and normalization 1 and 3 have a relatively high modularity. And so these pass the first threshold of selection. We then reassessed the between-subject reproducibility, the between-subject variability, in this case by calculating the coefficient of variation across 54 subjects. And so this is an example of this coefficient of variation matrix for each individual link in these connectivity matrices. And so what you can see here is that for Pearson correlation, normalization 2 and 3 that had high modularity have also very high between subject variability, so that is undesirable. Instead, normalization 1 is pretty good. For Euclidean similarity, almost all networks have low between subject variability. Normalization 3 is probably the one that has the highest. So in the end, we are kind of left with Pearson correlation with normalization 1, which is actually what the literature had already proposed, and Euclidean similarity without any normalization, because we don't, why apply a normalization if we can avoid it? So Euclidean similarity is attractive for sure for this purpose, because normalizations, the trending is altering the signal. And so we prefer not to do it if we can. Also, the Euclidean similarity approach has less statistical assumptions. It does not require a minimum number of data points to work, and it's not requiring that the data points of the two signals follow any kind of Gaussian distribution, while Pearson correlation actually would. And in addition, it just evaluates a different aspect of the signal. So it's evaluating the signal shape and its amplitude, which we know has a physiological meaning because it's what we use for kinetic analysis. It's what we use for kinetic modeling. Instead, with Pearson correlation, we are evaluating the covariation in the signal fluctuations. And unfortunately, the signal fluctuations have in kind of an unclear physiological meaning. I mean, they could carry a lot of non-physiological variance that is just related to the measurement error of the process. And so we moved forward with Euclidean similarity. And what you've seen so far are results for Euclidean similarity on the entire time activity curves. So for the entire 60-minute duration of the acquisition. And here you can see again the group average matrix, in this case, thresholded to keep only the 20% highest weights. But we can also separately assess the first 10 minutes, for example, or the last 20 minutes. And what are we going to get out of this? So the first 10 minutes are going to give us information more on the tracer delivery. And instead, the late time activity curve is going to just tell us something about the uptake of the tracer, and in this case, it's phosphorylation. We can also go more complicated, deriving these single subject metabolic connectivity matrices, not from the raw signals, but from the model-based time courses that we can derive from compartmental modeling, in particular of compartment 1, which is the time course of the concentration of the non-phosphorylated FDG, and compartment 2, which is the time course of the concentration of the phosphorylated FDG. And this is the matrix of the correlation among these 5 single subject connectivity matrices. And what you can see, for example, is that in the case of FDG, the full time activity curve and the last 20 minutes are giving us very similar readouts, because the amplitude is mainly driven by the tails in this case, but for a different tracer, it would be different. And we can also see, for example, that compartment 1 seems to provide information that are not contained in the other matrices, a kind of a unique information, because it has a lower similarity with the other matrices. And these are the corresponding full versions of those matrices without any thresholding. Now, let's quickly get to the second point. So with the same data set, now we have estimated the single subject PET connectivity matrices. We are going to estimate across subject PET connectivity using SUVR from the late time points, 40 to 60 minutes, and also from the kinetic parameters, K1, K3, and Ki. And so we have our subject series of participants, and we can estimate our across subject connectivity matrices. This is the one for SUVR, this is for Ki, for K1, and K3. And now if we relate them, what we see is that, of course, they have some similarity, but they are also kind of, there are some unique values to estimating each of these parameters. In particular, we can see that the K3 network is not so strongly related to the SUVR network. And then what we did was to compare the single subject with the across subject networks obtained in the same individuals, which we can do directly in terms of matrix to matrix, but we can also compare, like we did in this case, the hub nodes, so the highly connected nodes in these networks that we calculated as the nodes that have both a high degree and high eigenvector centrality. And so what you can see visually already is that there isn't a lot of overlap between the single subject and the across subject hubs. And in particular, if we assess the dissimilarity between these hubs, we can see that only the K3 network has some sort of a similarity with the single subject PET connectivity matrices. And this kind of echoes the independent findings that Sharna Yamadar has had with a completely different single subject molecular connectivity approach, because she's using continuous infusion in that case, but she's using Pearson correlation on normalized time activity curves. So again, we have that in her case, also the single subject connectivity hubs are more frontal, more in the frontal cortex. So again, with the question of how to validate, and in this case, we go back to the independent measures of connectivity. And so in terms of similarity to the structural connectivity template, both the single subject and the across subject measures have some similarity with the template, a dice of 0.3, 0.4. But when we relate the PET connectivity estimates to the functional connectivity obtained in the same subjects, we actually see that the single subject measures have a clearly higher match with the functional connectivity, which again doesn't mean the functional connectivity is a gold standard, because it's not, but it can point us in the right direction. And so finally, I want to say a few words on how we try to apply this single subject PET connectivity approach to a focal brain pathology. And so we again get back to how to validate, because one way to validate is potentially to look at how lesions disrupt these networks that we can estimate. And so this is the work that Julia has brought forward. So in this case, we are using a data set of 44 glioma patients acquired in Padova. And why did we choose glioma? Because it's a focal lesion. And so for a focal lesion, it's very important potentially to have an individualized measure of alteration, because lesions can be anywhere in the brain. So each subject can have slightly different networks. And also potentially in the view of in the future applying metabolic connectivity as a biomarker for gliomas or other brain pathologies. And also we wanted to compare this with another approach that is more frequently employed and that is more accepted in the literature, that is using just SUVR, so alterations in glucose metabolism in gliomas. And so this is the data set. These are the lesion frequency maps. You can see on the left the frequency maps of the tumor lesions, which means tumor plus the area of edema around it. And on the right, instead, the frequency of only the tumor lesions. And so you can see that they are mostly in white matter areas and mostly in frontal areas. And what we did here was to calculate these impairment indices. How does this work? So the individual metabolic connectivity that is calculated in each glioma patients is compared region by region against a group reference obtained from healthy controls, in this case from the WashU dataset. And so for each ROI, we are going to correlate its connectivity profile with the corresponding connectivity profile in the healthy reference. And that is going to give us an impairment index, because the impairment index in this case is just the complement to 1 to that correlation. So we have an impairment index for each ROI for each subject. And we compare it to what? Because we need to be able to say if this index is pathological or not. And so to be able to say that, we calculated the impairment indices with the same procedure also in healthy controls and built a healthy reference distribution, upon which we set a threshold at the 97.5th percentile. And so the subjects and the ROIs that have an impairment value that is higher than the 97, than the threshold, are going to be classified as impaired. And so for each individual, we have an impairment map of the regions that are above the threshold. And so what you can see here is that for low-grade gliomas, this is an example of one patient, the impairment map is typically only of regions that belong to the lesion area. Instead, for high-grade gliomas, we have impairments all over the place, not only in the same hemisphere, but also in the contralateral hemisphere. For SUVR, we calculated an impairment index more or less in the same way. So again, against a healthy distribution and using an impairment threshold of 97.5%. And so this matrix, which seems a little bit complicated, but this is the matrix of the impaired regions according to SUVR in green, according to metabolic connectivity in yellow, and according to both in purple. And so what you can see is that the purple is not really that frequently represented. This is also what we can see for each single network. So you can see that some networks are more impaired according to SUVR, and some others are more impaired according to metabolic connectivity. And so there seems to be a high level of complementarity between the informations that come from a network approach, single subject metabolic connectivity, and a local approach, SUVR. And finally, also, the metabolic connectivity impairment correlates with lesion volume, which is a known marker of disease severity. Instead, the SUVR overall impairment does not correlate with lesion volume. So, to conclude everything, I just wanted to stress that when we try to calculate single subject metabolic connectivity, molecular connectivity, PET connectivity, the choice of the similarity metric and the choice of whether to standardize or not standardize are extremely important, because we're going to get very different networks. We have chosen Euclidean similarity for a series of reasons, in particular the fact that it does not require any standardization. It allows us to play with not only the full-time activity curve, but also the early part or the late part, and also the single compartment kinetics that we can obtain from kinetic modeling. And it is based on similarity between signal amplitudes and shapes, and not on the fluctuations of the signal, which are of unclear physiological meaning. It can also be applied both to bolus injection data and to continuous infusion data, even though we have tested it only on bolus data so far. And we've shown that when applied to focal lesions, it can discriminate. It can show us both local and distant alterations, especially in high-grade gliomas, and it has some sort of an association with lesion volume. Now, with respect to the relationship between single subject and across-subject estimates, well, across-subject estimates, of course, we need to remember, are dependent on the parameter that we choose. So the across-subject SUVR metabolic connectivity is not going to be the same as the across-subject K3 metabolic connectivity. And we've seen that the overlap between across-subject and single-subject measures, at least estimated in the way that we did, is only partial. And so this tell us to be cautious when we want to interpret across subject measures as representative of the single subject networks. Also, the single subject networks in our case had a stronger agreement with independent measures of brain connectivity. However, validation. There isn't a gold standard for any of this. And so we need to keep pushing towards trying to understand the physiological meaning of these maps. And so, yes, this is my final reminder that we need to continue thinking about validation. We need to continue to be creative in ways to understand if these network measures, if these mathematical constructs that we come up with have any meaning from a biological standpoint. And so I thank you all for the attention. I hope I haven't bored you too much.
Yeah, you bored us very, very much, Tommaso. It was unbearable. No, thanks. I mean, I don't know how you could fit all of these in such a short time. It was very clear, very, very interesting results. So let's see. Do we have already some questions? Anyone brave enough to start the question part?
Yeah. Hi, Tommaso. Thank you for the talk. Really interesting. So I'm just wondering, so if you are interested in this within subject time series analysis, so I noticed that this dynamic PET image have different durations, right? That is optimized for this dynamic. You want to estimate this kinetic factors. But if you are interested in this time series analysis correlations, can you like bin it in a way that you have like shorter time, have higher time resolution? Will that increase like your ability to detect the correlations and the connectivity?
Sure, no, absolutely. I mean, what we have done in this case, of course, was to just interpolate the data that were already reconstructed onto a new uniform grid, a virtual grid, because then we can assess any measure. We can assess we can use Pearson correlation, we can use Euclidean similarity, we can use any of those. Now, I know that in some cases, people have directly reconstructed the data, going down to very short frames. That can, I mean, down to 1 minute, down to 30 seconds, 15 seconds, even less than that. Of course, that is more justifiable when you have a continuous infusion of tracer, because your radioactivity is going to remain high for the entire duration of the experiment. Instead, for the bolus data that we used, the framing that is chosen, and it's a framing that is chosen for kinetic modeling, but it's also chosen for very clear reasons, right? Because in the beginning, we have very fast events and a lot of radioactivity. And so we can go down to small frames of 5 seconds even, depending on the sensitivity of the scanner. Towards the end of the scan, instead, we have that the bolus, of course, is going down, and so the radioactivity is going down, so we have less counts. That's why we need to have longer frames to be able to still have counts and not to have very noisy data. And also the processes are typically much slower, at least for FDG now, because I'm mainly focusing, of course, on FDG, but other tracers instead have maybe a faster decay because they have a reversible kinetics, and so they're going down much faster, but still typically the last frames are 5-minute frames. So sorry, I just tried to give a kind of a convoluted answer, but I mean, absolutely, you can go down in tempo to have a higher temporal resolution, and especially with the new scanners, you have more and more the possibility to do it because you have the sensitivity to do it. You have less noise, but still, one needs to be very careful, because of course you can reconstruct data that are sub-seconds, but you need to have the counts to do it. Otherwise, you're just reconstructing noise. So that is something that everyone needs to be very aware of, and is dependent on the protocol that you are using and the scanner that you're using and so on.
Thank you.
Thank you.
It's a question from Gabriel.
Yes, thank you, Tommaso. It was a very, very illustrating talk. And perhaps it's a kind of a silly question, but what will be the effects of preprocessing on the data on both the group level and the individual from all?
Right. Well, of course, I mean, there's a kind of a basic level of preprocessing that we need to try to apply, right? So, of course, I mean, the preprocessing that is done during reconstruction and the attenuation correction and so on, and then some level of motion correction is always appropriate to perform. But if you're talking about the standardization, because of course then many people have tried to circumvent this issue of all the time activity curves sharing the similar shape with multiple different approaches for the trending the signal. So I've only shown a few of those because some people, I mean, in particular in Sharna's paper, she was using a special temporal filter that was denoising the data and at the same time trying to take into account the accumulation of the tracer during the continuous infusion of FDG and removing that, and so leaving only the fluctuations. And so, yeah, the point that I wanted to try to have come across is that each step that we apply to the signal may distort it. And so we need to be well aware of what we are doing, because each normalization, each detrending, each denoising is doing something to the signal. Especially another important thing, if we are doing denoising in time, so temporal filtering, we will then introduce correlations between time activity curves. And so our final correlation matrix is going to have higher correlations than expected because you have introduced this self-correlation, this auto-correlation in the signal that is shared across time activity curves. So,
but also the motion correction, right? Because then in the end, you are interpolating the images a little bit.
No, absolutely. That's why I said the motion correction, of course, needs to be careful. I mean, we need to balance, right? We need maybe a little bit of motion correction because, of course, motion happens during our scans. I mean, some people have, this was actually kind of a fortunate data set to some level, to some extent, because it was acquired at WashU, and most of those subjects actually wear a thermoplastic mask, which limits motion, but not in every case, actually. You can have still problems. So, I mean, some motion correction is typically required, but you're right, it's a potential confounder in this case.
Any other question?
Hi, I wanted to ask a question. Can you hear me?
Yes.
Yes.
I wanted to ask, so you compare to other structural or to other connectivity imaging methods. If, like, I mean, a wild guess in future research, what do you think will be the advantage of like PET connectivity compared to fMRI, structural MRI, for example?
Oh, that's a very good question. So, I mean, for sure, I mean, the company line that I could say is that because we have a clearer understanding of the physiology of the PET signal, we have an advantage over functional MRI, for example, functional connectivity networks, which have shown to be of use in many research areas and for many pathologies, but still we have I mean, after 30 years now, 35 years, we don't have a clear understanding of the we are very far from having a clear understanding of the physiology of the Bolt signal, of the resting state Bolt signal in particular. And so, yes, a more direct interpretability is for sure something that I think can be very important. But also the other message that probably I would like to have come across is that the multimodality, that's another kind of trivial answer, but the multimodality seems to be the winning way in many cases, because the structural connectivity is going to give you one type of information, the metabolism is going to give you another type of information, the fMRI is going to give you a mixture of other types of information again, and all can be useful in classifying subjects, all can be useful in individualizing treatment and so on. So we'll see what happens. I'm sorry if this answer is very vague.
It was a difficult question also. Sebastian. Okay, next question from Christina Erfurt.
Yeah, so maybe I want to follow up a little bit on this question, because in your talk you mentioned a couple of times that when compared to functional connectivity, you know, it looks a little bit different or it looks similar. So I think the problem we always face with the molecular connectivity, that we are not completely sure what's the ground truth, right? Because there's no way to really assess it. So I think therefore these validation studies that you presented very nicely today are super important. But also, like, usually I would expect that if you have, let's say, increased activity in the brain, you need oxygen, you need glucose, right? So in a way, there should be, at least in my opinion, some kind of a correlation, right? So the question I always get, like from people outside the PET field, would be, why would you use FDG if you can do it without any radiation, when you can do it with fMRI, basically? So my first question would be, how do you see the advantage of using FDG in connectivity over fMRI? And maybe the second one, in your matrices, you also see negative correlations. How would you interpret them in FDG?
Right. Well, thank you. Very, very interesting questions. Well, with respect to FDG versus fMRI, I mean, you're right. It can be difficult to push the cause of FDG, since it requires radioactivity. These types of approaches also typically require the entire dynamic acquisition, and so 60 minutes is a long time for subjects to stay in the scanner, while for fMRI, maybe with a 10-minute scan or even less, you can obtain your functional connectivity matrix, and it can tell you something about that. So, well, one point that I wanted to stress that was very quick instead on during my presentation was that at least with this Euclidean similarity approach on FDG, the tails, like I said, the tails, so the last 20 minutes were actually driving the entire similarity, and so potentially this approach could be done on a static acquisition, so on a subject that is undergoing FDG imaging. So of course you cannot remove that, but is undergoing FDG imaging for 20 minutes to get an SUV. And so if you just reconstruct those data with multiple frames, with I don't know, 3 or 4 or 5-minute frames, you can still get a network, a single subject PET network. But I know that is kind of circumventing the question. And another thing that I wanted to say is that I didn't stress this because I only talked about FDG, but of course these approaches could be potentially applied to any tracer. So PET has the advantage that every tracer is telling us something different about the brain, whereas fMRI is just a combination of blood flow, blood volume, oxygen consumption, and other things. And so potentially with different tracers, we could get different readouts that potentially could be useful for some pathologies. I'm sorry, I'm now forgetting the second question. The negative correlations. Yes. Yes, what I like about the Euclidean similarity approach is that by construction, it is a positive only matrix. So we are just, of course, we're getting it's 1 minus a normalized Euclidean distance. So the Euclidean distance goes in this case from 0 to 1, and it's 1 minus this value, and then we're rescaling it again. So it's just giving you a no similarity versus total similarity. Instead, Pearson correlation, like you were saying, it's giving us both positive and negative correlations, and so that's a problem because I mean, it's something that is more difficult to interpret, and so that's probably an additional point that I could have put in my list for Euclidean similarity.
Thank you. Thank you.
Thank you.
Okay, we have a lot of more questions. Would you like to stay 5 more minutes to try to?
Sure.
Yeah, Xine, you wanted to say something?
No, I just want to say that there's a question in the chat. So yeah. Okay.
Yeah. So first of all, we have a question from Jana Florea that essentially asks if you can see PET connectivity as have possible future use to assess the outcome of a treatment plan.
Right. Well, that's where we were trying to go with the glioma study. I mean, it was a kind of a first proof of principle that single subject PET connectivity can be used at the single subject level on a focal pathology like gliomas, but potentially it could be used for others. It's just that there was, on a focal pathology, there was the additional advantage of saying, okay, it's a focal pathology, so we prefer an individualized approach rather than a group level approach, even though indirect disconnection studies exist and have been done so many times. So I'm not saying that it can't be done, but so yeah, we definitely see it. It's probably a little bit early for that, but that would be the direction that we are interested in.
Great. Then I think Gabriel raised his hand first. So
thank you. So it's kind of a double question. It's related to what Christina and Sebastian's told. So the first one will be, do you know how will be the test retest reliability of it, or any data sets to test it? And the second part will be, does it has a circadian influence? Because I mean, it's related, like the time of the day where you acquire the data, kind of resting state might show some variations. Does that affect connectivity?
Very interesting questions, and we have never actually assessed the test retest reliability, even though we wanted to do it. And of course, it can be done, not specifically on this data set, but I know that OASIS 3, which actually contains some of these subjects, because it's also a dataset from Washington University. I know that dataset has at least 9-10 subjects that have test retest dynamic FDG studies, so it can absolutely be done. It would also be interesting to assess some within day variation, like you said, but again, it's not something that we've done already, but it could be interesting to put it in the pipeline.
Just ask a little note that actually the time of day effects, there are a lot of studies showing that even for a lot of neurotransmitters or like physiology parameters, that shows the time of day effect. So that's really using like MRIs or other any measures. So really that may be a problem you need to consider.
okay, we have another question. One thing just to point it out about, if you're looking for test retest data, you should ask Igor, because he has a lot of subjects, PET-MR, dynamic, and he has the raw data you can reconstruct how you want. So I think it's a good data set to use. We have another question from Michael Palner. By the way, nice to see you again. I remember you from Glasgow. So difficult question also, I think. But Tommaso, you can take anything. So with the speed of the FTG uptake following and IV bolus, does it make sense to do connectivity on dynamic scans from bolus injections? The first minutes will be driven by K1, and the uptake into neurons are probably quicker. What are your thoughts on that?
Right. So, well, our idea was to, I mean, of course, it depends on the metric that is used. In the case of Euclidean similarity, we kind of that's what we tried to explore. So we tried to do what? To separate the early part from the late part, precisely for this reason. So the early part was going to show us the tracer delivery and distribution much more, and instead the late part is actually the accumulation in the various areas, and the compartmental approach. So the model-based approach was to have kind of a more refined view of this. So to separate the delivery and washout, because, well, actually the first compartment is describing is influenced by all processes. So it's influenced by K1 that's going in, K2 is going out, and K3 is going out into the second compartment. So the first compartment is about the delivery of the non-phosphorylated FDG. And instead the second just rises and should be the phosphorylated one. So we try to take this into account with our methodology. I don't know if that answers the question. I'm not sure I understood exactly the question, but yeah, that was kind of in our mind to try to tease apart these elements, and of course it could be done for another tracer, but it would have a different meaning, because in that case the second compartment would be the binding and unbinding to a specific receptor, for instance.
Okay, great. I think Miguel is satisfied with the question. Okay, are there any other questions? Otherwise, I will ask a very quick one, just to make sure I understood correctly. So you said that in your case, you have a sort of virtual grid for your time activity curves. So like, how many time points do you actually have?
Right. So, well, the virtual grid is virtual. So you decide what is the spacing that you want. In our case, if I remember correctly, we were doing something like 15 seconds or 10 seconds across the entire duration of the scan. So it's just an interpolation process onto a new time framing, to have data that are uniformly spaced in time, and therefore to meet the assumptions of Pearson correlation or Euclidean similarity and so on, but without re-reconstructing the data, which would which would have to be done very carefully.
Okay, so there is some interpolation.
It's just a temporal interpolation. It's what we typically do in kinetic modeling, actually, because especially if we need to apply, I don't know, a convolution, we need a uniform spacing to be able to do that. Otherwise, the results are just going to be wrong. So okay. It's not altering the signal. It's just just changing the time grid. And so the signal is going to look exactly the same. It's just treatable because a non-uniform time grid is right. Problematic for operations.
Yeah. Okay. Any other quick questions before we close? Okay, I think, there will be many questions we could ask. The topic of validation in itself is enormous. So let's see if there's something in the chat. No, okay, just Michael reminding everybody to please click on the link in the chat and give us your feedback about the series, this talk, what you'd like to hear the next times. So we will take a break for the summer, and we will be back in September. So stay tuned, join our newsletter. And for those that will be at OHBM, please come and let's chat a bit about molecular connectivity and validation and PET imaging. We are happy to discuss. So thanks a lot. Thanks a lot, Tommaso. Great talk. And thanks everybody who asked the question. It was a very nice discussion.
Thank you.